Yeah, today we are going to discuss something about cancer stem cells. So cancer stem cells are basically small subpopulation of cells within tumor with the capability of renewable. They are self-renewable. They are having the capacity to differentiate. Usually cancer cells have mutations rather than cancer stem cells. So cancer stem cells are different from cancer cells. Cancer cells have been developed into cancer, while cancer stem cells have the chances to go into cancer. They can develop into normal cell as well as tumorless cell as well as tumor cells. So the differentiation and tumorigenicity will transform the cell into animal host. It can transform into animal host, and cell surface marker expressed by cancer stem cells can be CD44, CD24, CD33. 123, 133, and they can also express epithelial surface antigen. Why cancer cell can gain stem cell characteristic with the property of EMT, that is epithelial mesenchymal transition, EMT, that is shown by cancer cell only, and it is driven by certain kinds of protein which are inside our body. Some proteins are by the fetal time in our body, some are developed during the adult stage. So some protein which are responsible for EMT are SNA1, FEB, and TWIST. So these proteins increase the invasiveness in the epithelial cells so that they can undergo malignancy. So they are predominantly resistant to chemotherapy. So there is certain reason why they are resistant to chemotherapy, why these cells develop a resistance toward drugs. So the reason is ABC transporter cassettes. So we will discuss them also, and they develop resistance against chemotherapy, radiations, and origin come from adult stem cells by de-differentiation. So they de-differentiate. Basically, they de-differentiate, and cancer cell de-differentiate to develop into basically cancers. So adult EMT occurs due to malignancy, and a major protein which is responsible for malignancy is twist, TWIST. This protein is majorly responsible for developing malignancy in cancer cells. So we all have gene for TWIST twist from the fetal stage, but if this protein is expressed during adult time, then it can lead to cancer or malignancy. So if it is active in adult, so it is undergoing tumor or developing tumor. So cancer cells develop resistance against the drugs. We know that cancer cells are developing resistance against the drugs, otherwise they would be treatable. So why they are not treatable is that because they are developing certain kinds of resistance against the drugs. So which what is helping them to develop resistance against the drugs are ABC transporters or ABC binding acid, ATP binding acid. ABC transporters are membrane transporters which pump the toxins outside the blood. So, due to presence of these ABC transporters, the drug against the cancer gets pumped outside the cell, and the cancer cell develops resistance toward these drugs, and they can pump various distinct structurally molecules, unrelated small molecules outside the cell, and some kind of cytotoxic toxic drugs and dyes which can be pumped out by the ABC transporters. So but they use ATP at the expense of ATP. They pump out different kind of dyes and toxins outside the cells. So anti-tumor drugs can be pumped out and thereby resulting in low intracellular drug concentration. So definitely if ABC transporter is present in the cell and at the cell surface membrane, and drugs are inside the cell, so they will get pumped out, resulting in low concentration of the drugs inside the cell. So, anti-tumor drugs can be pumped out and resulting in lower concentration, and the elevated levels of ABC transporter can enable cancer stem cell to resist the current cancer therapies. So, these therapies do not work on cancer cells due to presence of certain proteins, which are ABC transporters, which are pumping out the toxins or pumping out the drugs, which we are providing. Actually, the chemotherapy which we are providing are being pumped out by cell through ABC transporter proteins. So the cells derived from cancer stem cells are subjected to further mutations. So we all know that cancer stem cell can develop into tumor or can be non-tumorous. So both type of cell can be formed from the cancer stem cell. It need not necessary that cancer stem cell will develop into cancer cells only. So the cancer stem cells can generate intraclonally both tumorogenic properties as well as non-tumorogenic properties. So cancer stem cells or cancer propagating cells are defined as self renewal and have the capacity of multipotency, means they can produce multiple kinds of cells from those cells. So we know that stem cells are able to form multiple kinds of cells. They can form total kind of cell, means whole as a whole, all parts of the body, or certain kinds of the body. So basically, cancer stem cells are multipotent, so they can form multiple types of cells. Phenotypes between the cancer initiating cells and cancer propagating cells may differ, and they can be dynamically changed. Cancer stem cells can drive tumor initiation and can cause relapses at the time of tumor initiation. They originate from either differentiated cells or adult tissue stem cells. They can also, cancer stem cell can display high plasticity, which are the changes, that is the phenotype, the changes in the phenotype and functional appearance. Such changes are induced by chemo and radiotherapy. So those cells can develop to plasticity. They can change the shape of the cell, the appearance of the cells. So tumor initiation can either be driven by differentiating cells or transformed tissue resident stem cells. There are two populations of the cells which have been identified in gastric ulcer: slow cycling cells expressing TF-MIST protein in gastric corpus, and leucine rich repeat containing GPCRs in gastric antrum. So the biomarkers which are present on these cells, we have already discussed. There were certain kind of biomarkers or certain kinds of markers or receptors which are present on these kinds of cells. So CD44 is one of the marker which is present in these cells. So it can be expressed in solid and hematological cancer, and expression is associated with increased proliferation, self-renewable, and metastasis. Uh, and CD44 and 166 cell population are able to form tumors fair. So basically, if these markers are present, so that is that cell is definitely undergoing tumor, and it is definitely going to form tumor. So CD44 and 133 expresses enhanced tumorogenic properties, and CD44, 24, and 45 may express breast cancer, and knockdown of CD4 will result in significant decrease and proliferation of migration of cancer stem cells. So there is a point, there is a catch, that if you catch CD44, so if you slow down the CD44, if we knock down the CD4 marker, then there is a chance that we can stop cancer to become malignant, and the proliferation of cancer cells can be stopped.